some.
Yes.
Yes, doctor.
Yeah, you can hear from my laptop, but not from this device. You know, we have a new.
Is that microphone? What's that?
I think there's a microphone
Yeah.
inside this.
Oh, the tablet.
Yeah.
What's behind that?
This one is for the camera.
No, it's for microphone like that.
Microphone.
But there's only one.
I don't know how to use this.
Since we have a
We can connect.
microphone, okay, use that.
Kita dah live ke kat YouTube?
Check.
Okay?
Kita dah live ke kat YouTube?
Not yet.
Dah.
Dah.
Wow.
Use my weapon.
Use the WebEx link directly. So we turn on WebEx here. The camera turns on, the microphone turns on by itself.
Oh,
And you just share the screen from there.
from
And
here.
yeah.
I need to connect with this, right? I
Don't
use
need.
my laptop and
No, you don't need to. If you join the WebEx directly, then the TV will directly join to WebEx.
oh,
Makes sense? Makes sense.
so you try to put the ID.
Yeah.
Forgot to bring the stand again.
So I will connect my laptop to see the TV like that.
So you'll be sharing your screen here as well, right? Okay. Good.
So the one that should switch on the audio maybe.
Mm.
Okay. Wow, so bright. Is it normal?
Hanis, I cannot hear you. Do you begin the presentation or what?
So can I have 5 minutes? My laptop is lagging.
Okay.
I can't hear you.
You're not in the camera.
Oh,
It's okay. Macam kita tiga orang. Dia memang tak muat sebab dia kat situ. Dia dah buka camera focus ni.
Actually, I want to use the
Phone.
my newer version of not newer version my new iPad, not really new, my iPad. So it can like move. It has this continuity camera, so it will move like when you talk, it will move here then.
oh yeah.
But it doesn't get connected now. I don't know why. So
So this is from this.
I can actually change here. FaceTime HD camera. But this is cool. I'll try to figure out how we can connect that camera to this so that we can just
Yeah.
use that one.
Even like the WebEx link, you can like directly open WebEx from here. You don't need to open WebEx on your laptop.
Yeah, but I still need to connect to here because I'm using OBS for streaming. Because that one doesn't have OBS.
Need to be the main laptop.
Yeah, to stream live, you have to have that stream key, which which you should not reveal or show to somebody else because somebody can reveal your live stream and I don't know all this, privacy stuff. Go to display setting.
Time display setting.
Anything yang boleh lift that up ke? I understand tu, asyik lupa je nak bawa. Your book ke, your...
You can even put like under the TV. There's like a place you can put the camera there.
Too low.
It's too low.
Too low.
The thing is group.
Then
What? TV screw ke?
okay.
Enggak lah, I need.
Dia boleh lift tapi itulah.
Tak payah lah, Mei, jangan pegang yang tu.
Kena ada screw dia.
Not something to lift this up there.
Yeah, it was. You don't need to share. Oh yeah. Yeah.
Usam,
Yeah?
Your mic has been kind of-
ready?
I think so. Yeah.
Assalamualaikum, Sam. Can you hear me?
Yes, now I hear you.
Your speaker.
Speaker tak ada.
It's very clear.
Okay. Speaker dah ada.
100. I think there's too many things open. Google Chrome, Microsoft Edge.
Oh lah.
Maybe that stuff.
Okay. Hello, can you hear me?
Yeah, yeah.
Alright. Wait. Screen.
You have to turn on. Okay, you turn on.
Oh, should I turn on the video? Should I?
Video.
For Usam.
Yeah, you have to share the screen for some because for some it's not.
She means like the front camera.
Yeah, you can.
Ah. Share. Okay. Sam, can you see my screen?
Yes, I see. But,
Okay.
yes. Now
Alright.
it's okay.
Good.
Okay.
Oh, bye.
Alah. Saya buat ni.
Check.
Oh, showing one. Vertical. Okay. Okay. Assalamualaikum and good afternoon, everyone. So today I will share. Okay.
That's okay. We can use our own screen.
Yeah.
Use our own WebEx or share the screen.
Why is doing only one?
Try again.
Cabut.
Set.
Keep on giving.
Shift and this.
Okay.
Okay.
Bismillahirrahmanirrahim. Amin. Bismillahirrahmanirrahim. So, Usam, can you see my slide, right?
No.
Share from here.
No.
Oh no.
No, I can't.
Okay. Right now?
Yes. Now I see.
Aduh.
Fine.
There is an option somewhere. I don't know. There maybe. On the bottom left corner.
Bottom left corner.
No, the 1, 2, 3, 4 maybe. I don't know. Just click this. Okay, that triple dot. Hide presenter view, or maybe you can swap because in Mac you can swap the.
Yeah. Okay.
Ah. Oh, macam mana? Okay, Assalamualaikum and good afternoon, everyone. So today I will share one paper that published in Microbial Pathogenesis journal. Okay, so this paper basically they talk about designing a multi-epitope vaccine using Toxoplasma ROP5, ROP7 and SAG1 epitopes and immunogenicity evaluation against the acute and chronic toxoplasmosis in BABLC mice. This paper from Iran that published in this year, 2025. Lagi. Oh my god.
If we just use WebEx, HDMI is taking load on your
Can you
Just disconnect this one. Yeah, sure. We just share on the WebEx screen.
Okay. Sorry for the inconvenience. Just I can see anything I need to.
Then I guess you guys turn on your camera so that
Oh yeah. I try to shut to close all the tabs. Okay. Bismillahirrahmanirrahim. Okay, so for the introduction, what is the Toxoplasma gondii? Okay, Toxoplasma gondii is a common parasite that infects almost all warm-blooded animals. Okay, how it can spread? It can spread through the contaminated water or food, eating undercooked meat, transplants, and congenital transmission from mother to fetus. Usually, uh, these infection silence in healthy people, but it's very dangerous for the pregnant woman because it can transmit to the fetus, and also for the immunocompromised individuals. This can cause brain, heart, or lung disease. Okay, so for toxoplasmosis or toxoplasma gondii infection, they rely on the infectious form. So in this parasite, they have three infectious form: trachozytes, bradizoid, and oocysts. And also sporozoite is inside the oocyst itself. Okay. For the trachozyte, is a rapidly replicating form that responsible for acute infection, and it spreads quickly and causes the tissue inflammation. For bradizoid, is slowly growing form, and it's found in brain and muscles during chronic infection. It can persist for life and resist treatment. Okay. For the oocyte, usually it's shed by infected cats, so it can survive in the environment, such as for soil and water, and it can cause the infection when it's ingested. Okay, so for sporozoite, inside the oocyte itself, it's very infectious form, filling the sporulated oocyst and released in the host gut after the ingestion. This initiates infection by converting the into trachozyte.
How long does it take for one cycle? No idea.
No idea.
Okay.
Okay, so far for treatment for the parasite, there's no effective cure for the chronic stage, and they still develop a vaccine for this parasite infection. Okay, for this paper, they focus on 3 antigen targets from the Toxoplasma gondii. SAG1, a major surface protein involved in the host cell attachment, ROP5, that helps that parasite survive by disrupting host defense proteins, and ROP7 trigger inflammation by activating signaling pathway.
So these three major protein in the parasite that they use for target the multi-epitope vaccine
for this paper.
So for
So
the
for the
objective,
objective,
okay, sorry.
So step one is a surface protein.
Yeah.
Yeah.
Cell attachment. ROP5 helps the parasite to survive by disrupting host defense protein, triggers inflammation by activating immune. Okay, these three proteins are they available in all of the 4 stages, 3 to 4 stages of the life cycle? Not sure.
To be honest,
I'm not sure, but in this paper they use 2 form
of
of
parasite.
parasite.
Two strain, two strain indicate two form of the
form
parasite.
Mm.
Okay. For the objective, to design and evaluate a multi-epitope vaccine targeted Toxoplasma gondii, immunodominance antigens, ROP5, ROP7, and SAG1, and to assess the multi-epitope vaccine protective efficacy against the acute and chronic toxoplasmosis in BLPC mice. So for the methodology, from that 3 protein sequence, they recreate in NCI, they predict the T cell and B cell using server,
and then they assess the entry, algorithmicity, and solubility. And then in this study, they optimize for the codon and in silico cloning to create the plasmid, the vector itself. And then they check for the docking, analysis, and immune simulation. That's one for the bioinformatic part. For the in silico in vitro part, they transform the plasmid in E. coli top 10 and transfect with the HEK293. And after that, they confirm and quantification of vaccine protein production using Bradford assay, Western blotting, and then they challenge the vaccine itself with the toxoplasma antigen. And then to evaluate the immunogenicity, they measure the antibody titers and cytokine assay.
The plasmid they use called as top 10 something.
No. Okay. I share you the result because I will use the PDF itself to share the result.
Husam, can you hear Hanis?
Husam, can
Yes,
you hear
Dr.
me?
Arkan.
Okay.
Yes,
Okay.
I hear you and Hanis and everything is okay.
Okay.
Top 10 E. coli is a popular strain of E. coli.
Yeah. This is the
That's,
equalist strain.
that's my
Or the
question.
PC
PCDNA.
PCDNA vector. Okay, this is the vector that they confront in silico. And then they will use this.
Can you zoom in?
Okay. Yeah, this one.
Amir, can you close the top of the cameras? Yeah. Full screen. Side by side. Yeah.
Okay, so basically from this plasmid, they cut and insert the DNA that code for the multi-epitope vector vaccine. And then after that, they transfer the HEK293 cells to express the protein. And then they will harvest the protein, and they will did the confirmation and quantification of vaccine protein production using the Bradford assay is to analyze the soluble protein. Meanwhile, for the Western blot, they did to check the insoluble protein.
They are using DNA vaccine.
Okay. I just based on the title, I thought it's epitope vaccine, but when I read the paper truly, this is DNA vaccine, but contain DNA that encode for the multi-epitope vaccine. So they get the epitopes, they predict the B cell and T cell using this protein, and then they use a certain HLA, murine HLA, and then they get the epitope. So this epitope, they reverse translation. Reverse translation, they get the DNA and then put this DNA sequence in the plasmid. So in the plasmid to amplify the sequence, and then they take this plasmid and transfer with the HEK cell. and then to express the protein.
Okay.
Okay.
To
To express the protein.
So the vaccine is
Yeah.
protein.
Protein. So protein is related. Yeah.
So it's recombinant protein vaccine. It's
not
not
Oh, no.
DNA.
It's what?
It's not plasmid DNA vaccine. Right. Because they purify.
Recombinant DNA vaccine.
That's really great.
Actually, I'm also not familiar. I just like learn this method. Actually, I have the reason why I chose this paper. Okay, fine. Okay, and then after they transferred this HPK293 cells,
Recombinant protein C vaccine protein production. So it's not the plasmid DNA vaccine.
I think recombinant DNA vaccine.
No, it's recombinant protein. They produce the protein here, right? Before they did the challenge.
Yeah. Okay.
They just express cells.
They have.
They harvest the
That's why they use the HEC293 cells here to produce the protein, and then they disrupt the membrane of the cells. Then they collected the protein, and then they did the Bradford quantification assay here, and then STS page and Western blotting to confirm the protein size.
protein
Yes.
size. Okay, and then okay, they also tested the vaccine. Okay, can I say recombinant DNA?
Protein.
But actually, based on my understanding, they not use that protein only for the validation. So that recombinant DNA can encode for that protein. Because for the in vivo experiment, doctor, okay, in vivo experiment, they have a mice parasite strain and antigen preparation. Okay, and this one for prepare for the TLA or toxoplasma lysate antigen. But in the immunization challenge, okay, they have 3 groups.
Oh, yeah. So okay. Understood now.
Maybe how I believe it.
You're right.
Yeah. So basically, I think I should jump to, because in this part they just okay, in this mice parasite strain and antigen preparation. Okay, in this part they explain on how they prepare the toxoplasma lysate antigen, and also they use 2 strain toxoplasma RH virulent strain. This is indicate the rapidly replicating stage. And the other one is PRU cyst forming strain. This one is representing chronic infection. So for the immunization part, they have 36 female mice, and then they divided these mice in 3 groups. So PBS is control group, and then empty plasmid, and also the recombinant plasmid that contain these multi-epitope vaccine candidates. Okay, and from these 3 group, they divided into the 4 subgroup. Okay, one is for the for the humoral stimulation, for the second cellular evaluation, and the third and fourth is strain challenge.
Can I ask what is RH and PRU?
Okay, RH and PRU is the strain for the Toxoplasma gondii parasite.
Oh, okay, so there are different strains.
Yeah, different strain, and then it's also different kind of infection. Like this one is for the RH virulent strain is the rapidly replicating stage in thalassocyte form.
Oh,
I
yeah.
Okay. And then for the PRU is a cyst forming strain. So this will apa?
Represent.
Yeah, represent.
Is there any reason why they only use female?
Then do not stay.
Right.
I don't remember, but I think I don't remember, to be honest. But there was something about
Parasite.
No. There was a reason for. I cannot recall it. I don't remember on top of my head.
But it's not specific for study lah. Every people specific for this email study.
I think it's quite general thought about that. I don't remember, to be honest. But
That means that they have specific reason why we choose.
that's why I said not specific. It's quite general thought. I don't remember whether there was a scientific evidence for it. I don't remember at all. But you know, it just clicked my mind when you asked that question. That if there is any reason.
Nothing to do with genetic like they reproduce female.
It says due to their more predictable and consistent immune responses. Male mice can exhibit behavioral issues such as aggression. So that could be
The genetic.
so female is more
Calm.
calm.
When they get the injection, you know,
Yeah,
like
easy to handle.
Okay, so this okay this this will the volume of these three groups in 100 microliter for the empty plasmid, and for the recombinant plasmid they have like 100 microgram plasmid for each. And then this vaccine injected through the intramuscular.
So what is MEVC? Containing MEVC.
Multi-epitope vaccine. So this plasmid contain the epitope that we retrieve in silico method.
So pcDNA3 is the empty plasmid.
Yeah, the empty
Okay.
plasmid.
The what do you call it? Template. There's a word for empty plasmid.
The empty plasmid.
No, it's okay.
No, I call it empty plasmid.
empty vector. Yeah.
Oh, empty
okay.
vector. Okay, and then they inject it with 3-week intervals, day 1, 21 and 42, and because the volume of the injection is 100 microliter, so they separate 50 microliter injection for each not a leg developmental leg muscle. And then why?
Just take vaccine, just shot one side.
I don't know, maybe like easy the vaccine to deliver. Okay. And then
Also, leg has more muscle, So there's less chance that you will injure a bone. Like quadriceps.
This person is muscle.
But this is intramuscular injection.
This person is
Parasite is
This parasite more present in brain,
in brain.
brain and
in brain and spleen. Brain and spleen. Okay, and then they have two other groups, for the challenge. After after 1 week, after 3 weeks. Okay, for the third group that challenge with the tacozide RH strain, after 3 weeks from the last injection, they will challenge with this strain, and then they will check for the survival rate and also for the symptom of the disease. Okay, for the PRU strain, okay, they IP challenge with a 10 tissue cyst, and 2 months later they harvest the brain homogenic. And then okay, because they check for the humoral immune response, so they check. Okay, they check the IgG, IG1 and IG2A antibody using the LISA and the cytokine assay and LPA assay for the for the vaccine induced memory and activation capacity. Besides, they also check for the quantification of the parasite itself. So they did real-time PCR using sample from brain and spleen. So for the result, actually in this multi-epitope vaccine, they use 12 peptides. 3 from B cells, 3 from MSC class 1, and 1, 2, 3, 4, 5, 6 from MSC class 2. This one. Okay, and then the figure 1 is figure 1. Okay. This figure 1 showed the 26 kDa protein from the expression with the HK cell.
Hanis, can I ask a question, please?
Yes, Z. Yes.
It's for the sample. I want to know what is the sample? You say the sample from brain and spleen. What's the type of this sample? It's the fluid from from the brain or spleen, or what's it exactly?
Okay, basically they take brain and spleen from the mice.
Yeah.
And then for the antibody titer, they just take the
From blood.
Sorry?
Blood samples for antibodies.
Yeah, but they from the blood they take the serum.
Serum, yeah.
Yeah, the serum.
Yeah.
Okay, and then like they did also the DNA sequencing for the recombinant plasmid, because they want to check either that plasmid contain the multi-epitope vaccine DNA. So the DNA sequencing confirm that the precise alignment of 250 residue MVPC fragment within the recombinant, this recombinant. Okay. So that means like that this recombinant plasmid successfully contain that DNA. So that's the result.
That's why.
Yeah.
Ah,
Okay.
they're checking the DNA sequence.
Good, because they use the recombinant DNA plasmid. So how they want to do that this recombinant DNA plasmid work? First, they send the recombinant DNA plasmid to the DNA sequencing. Second, they check for the protein expression. So the DNA sequencing right now, the result is 250 residue, and the fragment within the recombinant DNA. Okay. And then this result protein
Western
blot showed that 26 KDA protein.
Oh, this one dia ada lah.
This is the one that they are.
This is the protein.
The
The number
That's the
one
lattice.
is
protein.
protein.
What is that sound?
They some.
Yeah. All
Are
right.
you okay?
I get the answer. Okay.
Sorry, it's
But I want to go to the after 5 minutes.
Okay.
okay.
Salat al-Jum'ah.
Okay.
So I want to leave now. Can I?
Can. Okay. Bye.
Bye, bro.
Bye.
Thank you, Anis. Thank you for all of you. And salam alaikum. Say that to Dr. Asmeda.
Okay.
Good luck, Anis. Thank you.
Thank you. Okay, so right now they want to check either this vaccine, this recombinant vaccine is trigger the cell-mediated response or antibody mediated response. You know, like we have to this immune response in our body. So cell-mediated is to killing the infected cell. Meanwhile, the antibody-mediated response is like produce the antibody that can neutralize the infection.
Okay.
So they check for the humoral immune response. They check for the IgG isotype. Okay. First they find the IgG total will be 3. Okay, the reason why they use IG1 and IG2. IG1 is more to indicate the TH2 responses, more to antibody-mediated response, while IG2A is for the TH1 responses.
Can you repeat that again? IG1 is for?
IG1 more to cell-mediated response.
Okay.
IG2 IG1 is for antibody-mediated. IG2A is TH1 cell-mediated response.
Okay. Thank you.
Okay. So based on this result,
we can tell that first they check total IgG first. Okay, so we can tell that along the time, longer the time, the increase in IgG total in multi-epitope vaccine group compared to the placebo group in PBS. And then for the IgG1 isotype, to check the antibody mediated response, okay, the total of IgG1 also increase. Okay, the longer the time, increase the total. And then for the IgA also increase the total of the IgA in multi-epitope vaccine. However, if we compare to the IgA ratio with the IG1, IG2A showed more
more increase compared to the
IG1.
IG1.
So in
this
this
result,
result,
we can say that this
this
vaccine
vaccine
how to say
trigger
cell-mediated
cell-mediated
response
response
compared to
compared
the
to the
antibody.
antibody.
Okay.
Which is
And
better,
also,
right?
Yeah, because
because
cell-mediated can kill the cell directly.
directly.
Then
They
they also
also
did
did
like
the satu kinds
assessment.
assessment.
This
Is
result
this result also
complement
complement
with the
neural
response.
response?
Okay. In this one, they check for the
interferon gamma and
interleukin 4.
interleukin 4.
Okay, interferon gamma is
Interferon gamma is
cytokine for
cytokine for
TH1.
TH1.
Right now,
Right now,
IG to
IG
A, right?
right?
So this one is interferon
interferon
gamma.
gamma.
And then
And then
interleukin 4,
interleukin 4,
cytokine
for
for
the
TH2.
TH2.
Okay.
Okay.
So both
result
showed that
decrease the mRNA toxoplasma in
both like interferon gamma and interleukin 4. However,
mRNA.
because this one they did real-time PCR. This one is
Cytokine levels.
This is cytokine level.
This would be.
sorry.
Yeah.
Sorry.
Okay.
Hanis, they didn't use any positive control in this. It's just negative controls, a PBS and PCDNA3. There's nothing to compare the data with, if it's significant cell-mediated response or not. PCDNA is the empty plasmid. It's like a negative control. They want to see the reaction to the plasmid itself, if it produces anything.
So basically, we need some kind of
Like a positive control that shows a very high response, and then we can compare. Right, doctor?
You mean vaccine for toxoplasma as well, or what?
Something that produces an immune response, which we can compare the vaccine immune response with, because we don't know if it's significant or not. Like there's no positive control.
baseline.
Yeah.
Okay, this is a cytokine level, sorry, not the mRNA one. Okay, the cytokine level. And then the cytokine level in interferon gamma group for the vaccine group is higher compared to the interleukin 4. So this like complement the result that we get from here. Okay. And then the figure 3A, okay, this result showed the survival rate analysis in a mice with the RH strain, tachyzoids form.
So from this result, actually
all the mice
was
was
that.
but the vaccine group have like longer life compared to the
Because of the harvest or because of the
parasite. Because this one they check for the survival rate. And then this one they did for the cis number. This one they use the second strain, and then
so
the PBS group and the anti vector have a more number of cis compared to the vaccine. So that means like this because they did the microscopic observation for this assay. So in this result we can tell that the cis smaller compared to the
Smaller? No, lesser number, right?
less in number.
But
But
this
this
is
is
ah,
But
cis
did they do
company.
this after the mice died, or did they do this on which day?
After the this one after the
Mice died.
mice type, because you remember okay from kan we have like three groups, so from three each from these three group they divided into the subgroup,
Hmm.
so they have a four subgroup, right? Yeah, so for the group that challenge with the about the cis strain, only they check for
Oh, the
the
cis strain.
yeah, the cis
The
strain.
RH. No, not the RH. PRU.
So this survival rate analysis, so they include all the sub category. No, so what?
Only for the RH
All
strain.
RH
strain only.
strain.
Yeah,
The one is more
RH strain is
virulent.
virulent.
The one's more virulent. Betul?
But then PRU was the one with the cyst, right?
Yeah, the one with the cis.
Okay, so A is for the virulent one and B is for the cyst one.
Yeah.
Okay.
There's a bit different PBS and PCDC.
In which one? Cyst number?
No, the survival. Is it
PC, DNA, PBS. It's just one day difference.
one day different?
Yeah.
But I don't know it's accurate or not, because they did only like for 3 mice.
3 mice, yeah.
Yeah.
That's why I was like,
3 mice is too
no,
less.
for the
But three
survival
mice.
rate, usually we use this Kaplan-Meier analysis.
Ah,
What's that, Doctor?
the statistical analysis
The
method.
graph itself, you know.
Because they have the vaccine on day 21.
Day 42.
Yeah,
This one is the
survival
3 days.
analysis to estimate the probability of surviving a certain event over time. Probably because they just have 3 mice here.
3 mice only.
Yeah,
For each group.
Yeah, Kaplan-Meier, they will provide whether it's significant or not. The survival is significant. You know, Kaplan-Meier analysis.
The one that's also on the gene thingy that we see.
Every study is
Yeah.
mati.
Also, PBS and PCDNA groups have the same number of cysts. If you go down,
Is it 100%?
it looks too similar.
Too similar.
Like that looks.
So what do you expect of PCDNA and
No, I mean
PBS?
like suspiciously similar.
Because 6 is 6 like something.
Yeah, a little bit probably.
Maximum 6. The mice can have.
Probably.
I don't understand.
No, I mean,
But they did
But this is different group, right? The cis number analysis is different group, right? It's not for the
different group.
PRU.
PRU. But I may ask why the SBS and PCDNA have like almost the similar cis number.
Zain said that, for example, this one of the
Do they have like computerized software to calculate the cis?
Yeah.
They use a software.
Not software. That one like they
Yeah, I understand. Like a tool.
a tool. And then they standardize. You know, like the cis, how many cis you want to fight the mice. So maybe they can
Oh, that's why.
the
Oh,
similar
okay.
number.
Like that's the standard.
Yeah, the standard. They did mention in the protocol.
Can.
Okay, this is real time PCR. Okay, and then you know, like they want to check. After they introduce this mice with the vaccine, and then after they challenge with the parasite, and they want to check the quantity, or but in this paper they said like the toxoplasma burden, like the quantity of the toxoplasma itself in the cells. So for this real-time PCR, so we can say that
the
decrease
so
so
like
again
the more
more
CT
CT
value
value
is
is like
the
the
less
less
toxoplasma
toxoplasma
in
in
that
that
sample.
sample.
So we can say that
So
from
from
this
this
50
50
spleen
spleen
in
in
three
three
different
different
groups,
groups,
so
so
we
we
can
can
say
say
that
like
the
the
multi-epitope
multi-epitope
vaccine
vaccine
group
group
have
have
the
the
less
less
toxoplasma.
toxoplasma.
But if you want, if we want to compare between the RH strain, so we can tell that this vaccine more like
PRU.
effective
Effective
in
in
PRU
PRU
strain.
strain.
Yeah, use the one that
With the
Cis.
cis.
aggressive.
No, with the cis. The virulent one is the RH strain. Yeah, so this is the result for this one. And then for the discussion, Dr. Husam went to the
Prayer.
prayer.
Okay. So how many epitopes of vaccine in this?
12.
12.
Yeah, 12. 12, Doctor.
So they pull all the vaccine epitopes.
Mm-hmm.
Do they link all these epitopes?
Yeah, they link.
Did they provide information on that?
Yes, okay. That one because they all the bioinformatic result they put into the supplementary result. I share to you. Supplementary. Here. This is the 12
How big is that?
That's 154, 167.
wait, they did mention the size of the
So these binders are the linkers, or
no, this is only for the meat and then they link with
we can see
the
it on this. Okay,
wait.
okay.
Wait. The figure in here, the supplementary figure.
So why did you choose this paper? You said
Oh, because the last time when I attend the ICMBP conference, there one PhD student, but she already done with the work. She's still waiting for the graduation event. She did actually the way she explained is the same method that doctor want to use with the SLMS. Okay, but the difference is they take the peptide, they express in the E. coli. Same like this method because I asked about her paper. Did she publish her paper? She do not yet. So and then I found this paper similar to what she said. Now, so but indeed in wait, I want to remember assessment to okay, that experiment I don't remember the bacteria. Actually, for the bacteria protein, they have a specific protein. So they predict like us like in silico method. And then they express this peptide in the E. coli. And then they transfer to the cell, and then they harvest all the but the difference is they harvest all the surface all the protein, and then she mentioned about pull down assay. She pull down all the peptide, and then she did the MS analysis to quantify and to validate the peptide. And then she overlapped with the peptide that she did in silico part. Exactly like what you plan. Kan betul? 5-year works.
5 years.
Very good.
Ada
Dr. Wang
Sigmund.
Sigmund.
ITM.
That's the reason
I choose
to
this
express
because
the
it's
protein.
yeah. I never think about that one. Then actually I got this paper about peptide mixing, but I don't know.
If we don't express the bacteria, what's different if we, or because when they express protein, they express a lot of protein, or part of the candidate they also get the message.
The insulin, right?
In vitro, right?
No, I mean the one that is in the protein that they express. What's the protein that they express? Like the one that they get peptides, a lot of peptide that they express, or the
Oh, I forgot about one thing. Actually, she mentioned that she interact with something.
because what if we give this synthetic protein, then put in the cell, then put down acid, also get this.
What do you mean by synthetic protein? Recombinant? It's the same technology, you know. You can do it in the lab. But
Actually,
I
I
think it's quite difficult, quite challenging.
Actually, I miss that one critical word lah that she interact the protein with the peptide with something. I forgot lah. But I think that one can answer with the. I think that one can answer Ahmed. But can we conclude this paper first?
I think the line too long, right? Oh, the doctor already cut
No,
down
it's still there.
Okay. So I think
I think better
focus on this
focus on this first.
Office.
Okay je lah. Where is the key? No.
Kamu tak boleh share ke? Okay, for the discussion, for the discussion, the effective protection against Toxoplasma gondii requires the TH1 type immune response, mainly into the interferon gamma secretion compared to the TH2 responses. And then this response is important to clear the both acute and chronic infection. And then the in silico part that helps to identify the immunodominance epitopes from this Toxoplasma protein. And then actually in this paper they clarified this vaccine as a DNA vaccine. DNA vaccine. So, give long-term immunity by directly encoding the antigens into the host cells. And then the MBV-C protein around 26 kDa was successfully expressed in mammalian cells that confirm via the Western blotting SPS page. And then for the humoral response, vaccinated mice should increase total IgG and IgG isotype, favored TH1 dominance with either for Toxoplasma gondii control, high interferon gamma and low interleukin 4 levels were observed in vaccinated mice, confirming as a strong TH bias immune response. So for the conclusion, a multi-epitope DNA vaccine was successfully designed using the Toxoplasma antigens, and it induced the strong IgG, IgG1 and IgA, and also promote the TH1 bias immune response. So overall, the MBVC vaccine showed the promise in triggering protective immunity. Thank you.
still confused about the in silico part that you did. Like it's not clear.
Yeah, for me too. I was kind of waited for that, to be honest, because that's more related to our research, you know. But for the in vitro part, it's I mean currently we're not doing it. So
Maybe they have previous
Actually, I didn't put the in silico results. Actually, they have, but all in the supplementary.
I mean, obviously, if you have the in silico and in vitro result, in vivo result, so you want to emphasize on the in vivo result. Okay.
Do you agree with the vaccine?
This is then a vaccine.